looking through Michael's CV, he's been he's done some interesting things in his time. He did his undergraduate. You just look at his education, you'd think he's never been outside of the States, but when you look through his research experience, he spent time in Japan at the wicn Institute, he spent time in London doing research there, and even at the Max Planck in Tübingen in Germany. So he's doing some, working in some really interesting places, doing some really interesting work on attention. He did his undergraduate at Carnegie Mellon University in Pittsburgh, Pennsylvania. And then he got his PhD in neuroscience from the University of California. And currently, he is the assistant professor of optometry and vision science and neuroscience at the School of Optometry and Helen Wills Neuroscience Institute at the University of California in Berkeley. And I have to say, he's really enjoyed, from what he told me on Monday night over dinner, he seems to have enjoyed his time here in North Wales.
Absolutely.
And I think the weather has really turned it on for him. And having visited his area of the world, Berkeley, I can say that we've had a really lovely weather here, which has matched what you'd normally get in Berkeley, which isn't quite typical, but still.
Oh, it's exceeded it, I would say.
Yeah. So welcome, Michael, and let's hear what you've got to say.
Thank you. Thanks very much, Paul, for that lovely introduction. I'd also like to thank my host for my visiting scholar position here, Professor Bob Rafal, who's in Munich now giving a talk of his own, was unable to be here tonight. And also the Wales Institute of Cognitive Neuroscience and the School of Psychology here at Bangor University have been very gracious hosts and made me feel very welcome. Had some fantastic scientific interactions while I'm here. And also the people of Bangor and North Wales. I'm here with my family, including my mother-in-law, who's here to keep me honest tonight. And so we've been enjoying Bangor in the area and just had a fantastic time. And the people have been just terrific to us. So thank you for that as well. The topic of our talk today is how attention affects visual perception. It's well known that attending to certain locations or objects or features enhance our perception of those objects or features. Even though the pattern of light striking our eyes is exactly the same, our attention is enhanced when our perception is enhanced when we attend to a certain part of our visual inputs. And so in my laboratory, we're very interested in exactly how attention facilitates perception and what are the brain mechanisms by which this happens. I apologize in advance for some of the technical difficulties. There are a series of movies, and there might be a couple rough transitions, but we'll do our best to muddle through. I'd like to start with just thinking about the different ways in which we use the word attention in everyday life. When you start to think about it, there are really many different functions that we attribute to this single word attention. The first one here is something bright caught my attention. This refers to that aspect of perception that can be captured by unexpected events. So you may be very focused on me giving this talk, but if there's a bright flash over here or a loud sound behind you, even if you are trying to stay focused on whatever the task at hand is, your attention will be involuntarily captured by this unexpected event. The second notion of attention here is I didn't see you, I was paying attention to the game. This is the link between attention and perception, that we literally fail to notice very important events in our environment if we're not attending to them. And we have this illusion that's created by our brain that we are kind of taking in all of the visual information available to us. But the fact is a very small fraction of that information is getting into our visual consciousness, and attention acts as a gate or a filter to determine what it is that we're consciously perceiving at any given time. And this is a very important function of the brain. It would be paralyzing computationally to really process all of our visual inputs simultaneously at all the time. And so we select those parts of the inputs that are most salient, most relevant for us, and those get preferential processing in the brain. The third notion of attention here is I struggle to pay attention to the lecture. This refers to the effortful aspects of attention. So we all know how hard it can be to stay on task, whether it be reading a book or performing any number of cognitive functions. It's exhausting, and it really is fatiguing to exert our attention systems for a long time. And one of the unanswered questions in our field is, why is this so difficult? What's the limiting resource in our brain that makes it so exhausting to pay attention for a sustained amount of time? And finally, I don't even remember cleaning the table. I must not have been paying attention. This is a reference to the link between attention and memory. So just like attention acts as a gate on our perception, it acts as a gate on our memories as well. And we're much more likely to encode information that occurs at a place that we're attending compared to a place that we're currently ignoring. So there's also preferential access not only to conscious awareness, but also to long-term memory. So our ability to encode information is very much dependent on whether we're attending to that information or not. So the bottom line is that in common language, attention refers to many different kinds of mechanisms, and as neuroscientists and psychologists, we're interested in functionally defining these different roles for attention and understanding the different neural substrates that allow these actions of attention to take place in the brain. I meant to mention this at the beginning. We're a manageable sized group here, so if you have questions during the talk, please don't feel like you need to wait until the end. Go ahead and put your hand up. I'd be happy to take questions during the talk. Okay, so even though this is a free public lecture, I'm going to ask you to work a little bit during your time here. And specifically, I'm going to ask you to use your attention for a difficult task. The movie I'm going to show you is two teams of players, the white-shirted players and the black-shirted players. They each have their own ball that they're passing to each other. So I'm going to ask you to focus on the white-shirted team and count the number of times that they pass the ball to each other during this movie. Everybody ready? Okay, any volunteers? How many passes for the white-shirted team?
14.
All right, very good. How many of you counted 14? Show of hands. All right. How many of you saw the gorilla in the video? So it's a smaller number. Some of you did. I could hear a few giggles at the appropriate time. So for those of you who didn't, you probably have, or think that I'm maybe fabricating the gorilla in the movie. So I'm going to just replay the movie. But in this case, so when you watched it the first time, your attention was focused on a very particular aspect of the scene, that is the players with the white shirts. And part of what makes attention effective is that it actually suppresses information from locations that aren't being attended. And that's very useful in everyday life to be able to select relevant information from our environment. So now I'm going to ask you to watch the movie and just open your attention filters up and just see whatever you see. Don't worry about counting the number of passes. And I promise you this is the exact same video you've just seen. So not a very subtle visual event, right? A large animal acting in a threatening manner right in the middle of our visual scene, and it's very easy to miss it if we're attending elsewhere. And so this happens in real life all the time, not with gorillas, of course, but with very salient events that we simply fail to notice because we're not attending to them or we're not expecting them to happen. And we simply don't have the option in everyday life to rewind the tape and play it again to see what we've missed the first time around. And so we have this illusion that we are actually computing everything that's available to us in the visual environment. But we can miss these kinds of events all the time. And this is actually very relevant for the discussion of, for example, the use of mobile phones while driving, where we have the illusion there that we're driving perfectly fine as we're talking on the phone. And the research shows that talking on a phone, whether it's hands-free or not hands-free, it doesn't really matter. Having a mobile phone conversation while you're driving is similar to having had a couple of drinks in terms of the level of impairment of driving. And again, our brain is, I think, through evolution, very selectively designed to give us this illusion that we're taking the whole world. And we don't want to be always second-guessing ourselves and wondering what we're missing and being distracted by that. And so this kind of demonstration is very obvious because we can see the exact same set of visual inputs a second time. But in everyday life, this happens all the time. And we're usually blissfully unaware of all those visual events that we're missing. This notion that we fail to perceive events in the environment if we're not attending to them has been known to magicians for centuries before neuroscientists figured it out and started to study it. And magicians are masters of directing our attention to places where they want our attention to be and away from where we would want it to be in order to detect the trick. So I'm going to show you a magic trick now.
Hi, I'm Richard, this is Sarah, and we're going to perform the amazing colour-changing card trick with this blue-backed deck of cards. Now the idea is very simple. I'm just going to spread the cards in front of Sarah and ask her to push any card towards the camera.
Okay, let's see. I'm going to go for this card here.
Okay. Now Sarah could have selected any card at all from the deck, but she selected the card which is now face down on the table. And what I'm going to ask her to do is show us which card she selected.
Right, so the card that I chose was in fact the 3 of diamonds.
The 3 of diamonds, okay, excellent choice. That card goes back into the deck. Now I'm just going to spread the cards face up on the table. Give a little click of the fingers, and you'll see that Sarah's card here has now got a blue back. Not particularly surprising. What's slightly more surprising is all of the other cards have got red backs. And that is the amazing color-changing card trick.
So that was a really dumb trick, right? They took the cards off camera, switched it with another pack, and put it back. It's pretty obvious, right? And so this is an example of the selection by attention. So this was actually the very last frame of the video clip you saw. And the next thing you'll see is the very first frame of the clip. Okay, so again, we're going to watch this video again now with our attention filters wide open. And you'll see that even, you were sort of suspicious, looking for the trick in the video, and probably many of you missed these changes.
This is Sarah, and we're going to perform the amazing color-changing card trick.
This is a tribute to our last video here.
Now the idea is very simple. I'm just going to spread the cards in front of Sarah and ask her to push any card towards the camera.
Okay, let's see it. I'm going to go for this card here.
Okay. Now Sarah could have selected any card at all from the deck, but she selected the card which is now face down on the table. What I'm going to ask her to do is show us which card she selected.
Right, so the card that I chose was in fact the three of diamonds.
The three of diamonds, okay, excellent choice. That card goes back into the deck. Now I'm just going to spread the cards face up on the table. Do a little click of the fingers, and you'll see that Sarah's card here has now got a blue back. Not particularly surprising. What's slightly more surprising is all of the other cards have got red backs. And that is the amazing color-changing card trick.
Now, many people assume when they see this video that it really is a matter of just where are you looking. So we know our vision in the central part of our visual field has much better acuity and resolution power than vision in the periphery. And so the idea is if you were not staring at the cards all the time, but really looking at the appropriate part of the visual scene, that you would have a shot at noticing some more of these changes. And so a set of researchers have done what they call eye tracking experiments. So they have people watching this video, and then they have a camera on the eyes of the subjects as they're watching this video, and they're looking to see where are subjects pointing their gaze as they're viewing this video. So I'm going to show you an example of that now.
The University of Edinburgh have used sophisticated eye-tracking equipment to discover just where people are looking when they watch the famous colour-changing card trick. Each of the dots show the eye movements of a different person. The yellow dots show 8 people who missed all of the colour changes, and the red dot is from one woman who noticed Sarah's top changing from black to green. Hi, I'm Richard. This is Sarah. Straight away, we can see that faces attract a lot of attention, especially the eyes and mouth. Here people start to vary, with a few of the dots, mainly women, becoming fascinated by Sarah's rings, whilst the others focus on the cards.
Go for this card here.
Okay. Now Sarah could have selected. There's the first change, and no one seems to have noticed anything. Which is now face down on the table. What I'm going to ask her to do is show us which card she selected. Remember, the woman shown in red noticed the change, but interestingly was looking in exactly the same place as all of the others. It's only now that she starts to behave differently to other people in the group, flipping between the two presenters. So what does this tell us? Well, this type of research suggests that although everyone was looking in the same place, and therefore the same images were entering their eyes, most people were selecting what they wanted to see, focusing on the trick rather than the colours, and so they missed what was happening right in front of them. And that is the amazing colour-changing card trick.
All right, so as you can see from this demonstration, these effects of attention on visual perception actually have very little to do with where our eyes are directed at any given moment. There is a relationship between where we're looking and where we're attending, but it's not a necessary relationship. And it's perfectly possible for us to look at one part of our visual field and direct our attention elsewhere. This is a phenomenon called covert attention, and it will be a topic of a lot of the research I'll present to you today. Okay, so now I'm going to ask you to do another attention task, What you're seeing here is a pair of images that are alternating back and forth, A, B, A, B, A, B. And they differ in an important respect. So I'll give you a few seconds to be able to search over this space with your attention spotlight and see if you can notice the difference between the two scenes. OK, how many have spotted it? Yeah, a handful. All right, so I can make the task much easier by directing your attention to the appropriate part of the scene. So if I say, direct your attention to the bottom of the base of the wing of the airplane, then it should become obvious what's differing between the two. Again, not a subtle difference at all. An engine of an airplane appearing and disappearing is something you would think would be pretty noticeable, but in fact isn't if you're not attending correctly to it. And so the process of me directing your attention there is in utilizing one of the two systems of attention in the brain. And these are called endogenous attention and exogenous attention. Endogenous attention for being generated from within. So if I tell you to direct your attention to the base of the airplane wing, you take that information coming through your auditory system, your eardrums, and your brain translates it into a location in space that you can then direct your visual attention to in order to be able to see the difference. So this is called endogenous because it's generated by you. You make a voluntary decision about where you're going to direct your attention based on what you're trying to accomplish. In this case, you're trying to detect the difference between the two images. OK, here's another example, the same kind of phenomenon. Again, the two images are a pair that are alternating back and forth with one important difference between them. And so in this case, I'm going to direct your attention through your exogenous attention system with my pointer. OK, so just that little red light, a very small change in your visual inputs, gives you a very different conscious experience of this film. And so it's exogenous attention because it is the kind of attention that is drawn by a loud sound or an unexpected flash in the environment, for example. So in this case, whether you wanted to or not, some of your attention resources will be drawn to the red light. So it's an involuntary exogenous from without type of mechanism. And as you can see, both of them have very powerful effects on our visual perception. So these effects of attention on visual perception are just one example of a basic fact about our visual perception, which is that it is a very active constructive process. So we typically consider our perception to be a passive recording of the environment. We consider our visual system to be basically a camera, and we point our eyes around and we gather information, and it tells us what's out there in the world. But in fact, the metaphor of a camera is actually a very good metaphor for our eyes. Our eyes have pinholes, they have a lens, they have a sheet of light-sensitive material in the back. But the metaphor completely breaks down once we get beyond the eye into the brain. And a great deal of our visual perception is actively constructed by the brain. And we use our attention, our expectations, our whole life experience with the visual environment in order to be able to build, to make inferences about what's out there in the world. We're not just simply passively recording it. And so I'll show you a number of examples of perception as this active constructive process. Here's one of them. These two images are exactly the same, except one has been inverted with respect to the other. And probably most of you see this very clearly as two craters within two mounds, and you see this as two mounds at the bottom of two craters. In fact, the visual input is completely ambiguous with respect to whether it's concave or convex at any given place in the image. It's our visual system that's making assumptions that the direction of the light source in this photograph is probably coming from above, which is a pretty solid assumption in everyday life. The sun or artificial lights are usually coming from above. And so without really kind of checking in with us, it just decides that this ambiguous figure is going to be perceived as a crater inside of a mound, and this one as a mound inside of a crater. But in fact, there's nothing in the image itself that determines whether it's one or the other. It's a perfectly ambiguous image. And so our visual system is disambiguating it based on some educated guesses about the nature of light and the nature of shape from shading in these images, and giving us a very unambiguous visual perception, which turns out to be the correct one in most situations. Here's another example. So you see that this is a checkerboard surface with a cylinder, and you can see that the two squares labeled A and B appear to be very different in terms of their luminance. In fact, they have exactly the same physical luminance. If you were to measure the number of photons coming off of square A with a light meter, and then measure the number of photons coming off of square B, they would be exactly the same. And this can be demonstrated very easily just by adding something that disrupts the visual context in the scene. Okay, so these two are exactly identical except for the gray bars. You can also, if you make a tiny pinhole, close one eye, make a tiny pinhole over the other eye so that you're just viewing one of these squares in isolation without the surrounding, you can verify for yourself that these two have, in fact, exactly the same physical luminance. So at one level, this is a visual illusion. A and B are, in fact, exactly the same in terms of their light properties, but we perceive them to be very different. So in that sense, our visual system's getting it wrong. It's not giving us an accurate representation of what's out there in the world. In a deeper sense, though, our visual system is getting it right in that we are taking into account the fact that this cylinder is here and that light is coming from here, casting a shadow onto B. So the only way that this square could have the same luminance as this is if it, in reality, had a lighter shade of pigment or paint on it. And so really, ultimately, what we care about is not the particulars of the lighting conditions. We care about the true reality of the objects in our visual environment. And so our brain does what's called discarding the illuminant. And so it takes into account these contextual properties of this image to give us what is really the right answer, that B must have a lighter shade of paint in order to be able to produce the same luminance as A, given that it's in a shadow. And so we unconsciously constantly correct for these sorts of changes in lighting conditions. So if we look at an object under fluorescent light versus incandescent light versus sunlight versus light on a cloudy day, the actual photons that are being reflected by the object are radically different. But we effortlessly are able to perceive it as the same object across all these different lighting conditions because of this active constructive aspect of our visual perception. Another way in which we actively construct our perception of the world has to do with the way that our visual system deals with changes in our eye position as we're viewing the world. So in natural vision, we move our eyes about 3 times a second. And imagine if we just put a camera at the very center of our retina and just filmed whatever was coming in. It would be this nauseating, disorienting video where every 300 milliseconds, every third of a second, we would be getting a completely different picture of the world. As we move our eyes, a totally different image falls on our retina. But of course, we don't perceive the world that way. We perceive it as this continuous, seamless representation. And so there are active mechanisms in our brain that sew together these little snapshots of the world that we get on our retina. And a big part of that is that the same system that executes the signals to move our eyes tells our sensory apparatus about that eye movement that's coming up. And so our sensory apparatus can predict the eyes are going to shift in a certain direction and angle, and it will then basically anticipate what that change will mean for the visual environment, and allows it to sew together these different snapshots of the world. And this can be very easily demonstrated if you just cover one of your eyes with your hand, and then take your finger, just very gently displace your eyeball a little bit. And the perception you'll have is that the world is moving. Your eyes are moving just possibly a millimeter or less than a millimeter. But your perception is that it's actually the world that's moving. And the reason for that is that nobody told the sensory apparatus that an eye movement is going to happen. And so it interprets movement of the eye in this case as movement of the world across the retina, as actual motion in the world. And of course, when you make an eye movement in normal vision, it's a much more radical change to your visual input. But because the oculomotor system, the system that generates the eye movements, has sent signals to our sensory apparatus to inform it about this, we are unaware of the eye movement, and we just simply take in the world as this continuous, seamless visual experience. So this requires a lot of neural machinery in order to be able to correct for all of these radical changes in our visual inputs with eye movements. Okay, so hopefully I've convinced you about some of the active constructive properties of visual perception. And a big interest in my lab and many other labs in the field is how does this actually happen in the brain? What are the computations that take place in the brain that give us the very rich visual experience that we have? And so we'll start with a basic description of the anatomy of the visual pathways in the human brain. All right, so I'll just turn the lights off for a second. Here's a side view of the brain with the eye looking at an image in front of it. So light enters the pupil, of course, and is focused by the lens onto the retina. And then the retina has light-sensitive cells that transform the light patterns into electrical signals. Those electrical signals are conveyed into the very center of the brain, a structure called the LGN, or lateral geniculate nucleus. Geniculate because it's in the shape of a knee, so similar to the root of the Latin word genuflect. There are other neurons in the LGN that in turn send connections all the way to the back of the brain. So it's one of the quirks of evolution that the part of our brain that processes visual information is as far away from the eyes as one could get. But that's the way we're set up. And so the whole surface of the brain is covered by a thin sheet of cells called the cerebral cortex. And it turns out that the back of the brain, the back of the cortex, is the part of the cortex that's specialized for visual processing. Here's another view now from the bottom of the brain. So now we see the two eyes and the optic nerves carrying information from the retina to the LGN on either side. One of them's here and one of them's here. And again, the LGN send axons all the way to the back of the brain, to what's called the primary visual cortex. So it turns out there are many visual cortical areas. The primary visual cortex is the first one in the chain of processing. So that's where the term primary comes from. Okay, so now we're going to look at the projections in a little bit more detail. So this is now a slice through the brain, of course a cartoon. The two eyes are viewing locations in both the left and the right visual field, so to the left and the right of wherever gaze is directed. And so if we track the processing of an object in the left visual field, it will illuminate the right side of both eyes. And it turns out that the connectivity in the brain is different for objects in the left visual field and the right visual field. So in the left visual field, these neurons in the retina, on the nasal side, meaning closer to the nose, as opposed to the temporal side, closer to the temples. So red is encoding information in the left visual field here. So these neurons actually cross over to the other side of the brain, where they terminate on the lateral geniculate nucleus here. Neurons in this eye that are processing the same location in the visual field don't cross. They stay on the same side and terminate in the same lateral geniculate nucleus. And the pattern is exactly symmetric on the other side. So the right visual field illuminates the left side of both eyes. On this side, the left side, the axons, the connections from the retina do not cross. They stay on the same side in the LGN. And these do cross. And so what you have now at the LGN is that both the left and the right LGN receive input from both eyes. But they differ with respect to what locations are represented. So the right side of the brain is representing locations in the left visual field. The left side of the brain is representing green locations in the right visual field. So this pattern is called a contralateral organization. Contra simply meaning against or cross, lateral meaning to the side. So the left side of the world is represented on the right side of the brain. The right side of the world is represented on the left side of the brain. And then these neurons in the LGN that send connections back to the primary visual cortex maintain this contralateral organization. So even in the cortex, you have the left cortical hemisphere representing locations in the right visual field, and the right cortical hemisphere representing left visual field locations. So it turns out this is certainly a level of detail to describe this, but if we're interested in really understanding the neural substrates of visual perception, we would like to know in great detail exactly how each visual location is represented in the brain. And it turns out that our first understanding of the layout of the visual field in the brain representations came in no small thanks to these three gentlemen, none of whom were scientists. They were all developers of weapons and munitions. So Whitworth, famous for the rifle he designed, and these two guys developed new and improved, so to speak, bullets. And this turned out to be important in the history of visual neuroscience because as a result of their efforts, there was a dramatic increase in the speed at which bullets left weapons as they were fired. So this is time on the x-axis, muzzle velocity on the y-axis. And you can see from the Franco-Prussian War in 1870 to the Russo-Japanese War at the turn of the century, there's a dramatic increase in how fast bullets flew out of guns. And what this meant is that for the first time, bullets would, when striking a soldier's head, pass all the way through the head, cauterizing the wound as it went. And so it made very circumscribed, well-defined lesions in the brain. Whereas before that, the bullets were going so slow that they made big, messy, ugly lesions in the brain. And so this turns out to be useful because there was a doctor who was employed in the Russo-Japanese War by the Japanese government. His name is Tetsuji Inoue. And it turned out the system the Japanese government had was if you experienced any kind of visual field loss, any blindness in your visual field due to injuries sustained in combat, the pension you were entitled to was proportional to the amount of visual field loss you had. And so this military physician was assigned to measure the amount of visual field loss in soldiers and then tell the government how much pension that they should be receiving. But being a good scientist, he not only measured the amount of visual field loss, but he measured the location of the visual field loss and correlated that with the trajectory of the bullet through the back of the brain. And so he was able to make a detailed map of the relationship between locations in the world and the representations of those locations within the brain. And that's how we obtained our very first visual field maps in the brain, was thanks to his efforts. So these days, we don't use those methods quite so much to understand brain organization. We're more likely to use machines such as this one. So this is a magnetic resonance imaging scanner. There's one of them just across the way in the School of Psychology. And Bangor's at the forefront of research with using this technology, and Paul in particular is an expert on this technique, so set me straight if I say anything wrong. In particular, a very powerful method is functional magnetic resonance imaging. So MRI can be used to obtain structural images of the brain, but it can also be used to measure functional aspects of the brain. That is, it can give you an idea of what parts of the brain are most active at any given time. And so as experimenters, we can set up patterns of visual stimulation or tasks that elicit patterns of brain activity that we can then measure non-invasively in the brains of subjects who are inside this device. And so I won't get into the details of the technology, but basically using these methods, we can obtain movies over the entire brain of exactly which areas are active at any given time. Using these sorts of methods, basically neuroscientists, visual scientists, present visual stimuli at different visual field locations. So subjects maintain gaze in the center of their visual field, and they stimulate different parts of a screen, a computer screen that the subjects are viewing. This illuminates different parts of the retina, and then as I've shown you, the retina carries that information via the visual pathways back into the visual cortex. And what has been found is that there's not just one map of the visual field along the surface of the cerebral cortex. There are many, many maps. Just a handful of them are shown here. And they're named V1, V2, V3, essentially in the order of the processing in a visual, a hierarchical sequence of processing steps. And so adjacent locations in the world will illuminate adjacent locations in the retina, which will then activate adjacent locations in these maps. So each color here represents a full map of the visual field in this contralateral organization. So what we're seeing here is the left hemisphere. Here's the back of the brain. So the eyes would be way up here. And so all of the V1, for example, has every location that's visible to us in our right visual field. The other hemisphere, not shown here, would be the right hemisphere that would represent all the left visual field locations. And then this map is replicated again and again in these different numbered areas. So I'll show you in more detail now exactly how the visual field is laid out on the surface of the brain. So this is, again, the back of the brain. This is a sulcus, so one of the valleys in the brain that's been opened in this drawing so you can clearly see the walls in the bottom of this valley. So this is the left hemisphere. We're basically, if you imagine taking the two hemispheres of the brain apart and looking at the internal surface, that's what we're looking at here. So left hemisphere will only represent right locations in the visual field. Here's the center of gaze. Anything in the left visual field will be represented in the right hemisphere, which is not shown here. This is called the horizontal meridian. It's basically from the center of gaze directly out to the right. So 3 o'clock, if you consider this to be the hands of a clock. That's represented along the floor of this sulcus here. Here's the lower vertical meridian. That turns out to be represented on the top face of this sulcus. The upper vertical meridian along the lower surface here. And the very central visual field location, so right at the center of our gaze, is represented in the very back of the brain. And you can see that there's not a lack of proportionality between visual field locations and brain locations here, whereas the central visual field is represented by a very large portion of the cortical surface. And all these other locations are represented by this portion here. So there's an expansion of the cortical territory that's devoted to processing the central visual field. And this is a big part of why our central vision is so much more sophisticated than our peripheral vision, is that there's simply more neurons, more circuits in the brain that are devoted to processing information from the central vision. So the consequence for this is that we have a very distorted map of the visual field. So if this would be what you're looking at with the crosshairs being the center of gaze, this is how this image would be represented on a flattened representation of your visual cortex, where this would be the central part of the visual field, massively magnified, and this would be the peripheral part, which is massively compressed compared to the center. So if you were to actually make a movie of brain activity in this primary visual cortical area as people view different visual scenes, you would see this kind of distortion, a magnification of the central visual field locations. And one of the mysteries in our field is if this is the world as objectively exists, and this is how we represent it in the earliest parts of our visual system, how do we ever get back to an experience of the world that resembles this as opposed to this? How do we transform this very distorted view into a realistic, veridical representation of the environment? Okay, so it turns out all that we've talked about so far are a very small handful of the many, many cortical areas in the brain. So here what you see at the bottom is RGC, retinal ganglion cells, the lateral geniculate nucleus, and V1. And so this is the part of the visual system that we've discussed so far. And this is actually somewhat out of date at this point, but this is a sample of the large number of areas in the cerebral cortex that have some role in processing a representation of the environment. And so you can see that in V1, there's already a map of the visual field, and there are neurons that respond to visual stimulation. So what in the world are all of these other areas for? And I and many others would argue that they are a big part of this constructive inferential aspect of vision, that the part of visual perception that goes beyond what is given us by our retina, all of these influences of, for example, the effects of attention, the inferences that we make about the direction of light or visual context, all of these active constructive aspects of vision are due to these areas making very sophisticated computations about the likelihood of different events in the visual world and sending signals back down to the earliest parts of the visual field to be able to influence how information is processed in those early visual areas. And so, for example, in the gorilla video, these areas are the ones that are encoding your goal of trying to count the number of passes in the white-shirted team. And so they select those parts of these early areas that represent white shirts and people wearing white shirts, and they suppress those that represent other aspects of the visual scene. And so as a result, you're able to select your attention very effectively and filter out those parts of the environment that are not as beneficial for you at any given time. Okay, so now we're going to look at a more microscopic level at the organization of the cerebral cortex. So both of our hemispheres are covered by this thin sheet of neurons, shown here in kind of a purple. This is just a result of the particular tissue stain that was used in this thin section of a brain. And so basically you can imagine just looking at the face of this, for example. And so what you see here are the gyri, which is the ridges of the cerebral cortex, and then the sulci, which are the valleys in the cerebral cortex. And you can see it's very convoluted, and this is really evolution's solution to being able to give us a very large cerebral cortex for lots of computational power without making our heads so massive that they would endanger women's lives in childbirth. Literally, this is sort of a compromise that evolution's had to make. And so we have a much more convoluted cerebral cortex than many other mammals who don't rely as much on their cerebral cortex for survival. So within this, the gray matter, so the gray matter is this region here. It's called gray based on without any tissue staining, just the color of the brain is gray here because of the large number of cell bodies within it. Underneath it is the white matter, which is white because it's got a large number of axons or connections between neurons that have a fatty sheath around them, and so the fat appears white in the tissue section. And the intricacy of the connections within a small patch of cerebral cortex is just astounding. So within a cubic millimeter, so 1 by 1 by 1 millimeter, there are on the order of 100,000 neuron cell bodies, an uncountable number of dendrites, really, and then local axon branches. So these are the output connections of neurons. So within each cubic millimeter, there are literally 3 kilometers of axon branches. And this is true all over our cerebral cortex. The white matter are axons that connect parts of the brain that are far apart from each other. So maybe this part of the brain projects information here through the white matter. So when we do experiments with fMRI to understand the brain organization, we are primarily interested in the gray matter because this is where the neurons really are connecting with each other and where all the interesting computations are taking place in the brain. And so we develop algorithms to be able to segment that part of the gray matter. So this is now an anatomical MR image through a section of the brain. And so the green boundary is the outside of the brain, and the yellow boundary is the boundary between the gray and the white matter. And you can see actually fairly little of our cerebral cortex is actually gray matter. A lot of it is the white matter that involves these long-range connections. But the gray matter is really where the action is in terms of understanding representations in the brain. And so that's the part that we focus in on. Okay, so now I'm going to tell you about some experiments that I was involved in where we were interested in finding maps of the visual field in the brain, but not maps that simply respond passively to sensory stimulation, but rather maps that encode the location of our attention. So I told you about covert attention. You can have your eyes directed towards a certain part of the visual environment, and you can direct your attention like a spotlight in many other locations. And so we're interested in this process in the brain. How do we direct our spotlight to certain locations? How does that take place in the brain? And what consequences does that have? How does that help facilitate perception at the attended location? And so the experiment we did involved subjects looking at this central point without moving their eyes, and then directing their attention around this circle systematically. So this loudspeaker is a schematic for a spoken number that the subjects heard. So they heard a number 1 through 8. Each number designated one of these segments. And so, for example, they would hear the number 7, direct their attention here, and then this target would be shown on half of the trials, and not on the other half. So the targets here are shown at a high contrast so you can get a sense of what they look like. When people actually participated in this experiment, the targets were very low contrast, extremely difficult to see. So they failed to actually detect some of them. And the reason for that is that puts demands on the attention system. If it's very difficult to see, then you really have to exert a lot of attentional effort in order to be able to perceive the target. And so on each trial, they would hear the number, a target would be presented or not, and then they would push a button indicating whether they had perceived the target or had not perceived the target. And on the next trial, the attention would move on to the next location. So over time, attention was sweeping around the visual field in a periodic manner, just like the hands of a clock. The reason that there are stimuli shown to all of these different segments is because we were interested in just looking at the effects of endogenous attention, sort of pure attention signals without any contamination, if you will, from the sensory processing in the visual stimulus. And so you can see if you just look at this, on each trial, there's a random pattern of stimuli. So there's a 50% chance of one of these grading target stimuli being shown in each one of these segments. But the subjects are only ever attending to one of them, and they're ignoring the other 7. And then on the next trial, they move on to the next one and ignore the other 7. And so there's no systematic pattern to the visual stimulation, but there is a systematic pattern to attention, in that attention is traversing the visual field in this periodic manner. And so let's think about what the pattern of activity would look like in the brain while subjects are performing this task. So this is now just cartoon data. If we consider these to be 2 visual cortical areas that each have maps of spatial attention signals. So this is in the right hemisphere. So remember the contralateral organization. This will only represent left visual field locations. So as attention moves through the right visual field, there's no activity here. The activity would be in the right or the left hemisphere. And then as attention enters the left visual field and moves up, there's going to be this traveling wave of activity that moves across these areas. So for every single location, I forgot something very important. What we're looking at here is a flattened representation of the cerebral cortex. So remember, we segment out the gray matter part of the anatomical image, and then we computationally flatten it out. This is one of the movies that I was unable to show you. So what this is now a 2-dimensional representation of this convoluted cortical surface, where the dark areas indicate the valleys or sulci, the light areas indicate the ridges or the gyri. So imagine just taking a portion of the outside surface of your brain and literally just flattening it out into 2 dimensions. And then we're viewing the functional activity superimposed upon this anatomical flattened image of the cerebral cortex. So again, as attention moves through the left visual field, we see these traveling waves of activity in the right hemisphere. And so it turns out that before we had conducted this experiment, there were many visual cortical areas that had been defined simply based on passive visual stimulation. So subjects would just stare at a screen, different visual locations would be stimulated, and through that people were able to derive maps of V1, V2, V3, et cetera. And then that had pretty much reached a certain portion. So there were a number of areas that were defined, and it seemed as though those were all the areas that could be defined using that method. And it was only through the introduction of this attention task that we were able to discover 2 new areas. And these areas respond very poorly to passive visual stimulation. So if stimuli that are presented that have no behavioral relevance to us, that we don't care about, those fail to activate these areas. It's only when you direct attention to a certain location that these areas indicate where your spotlight of attention is being directed towards. So these are where these areas are located in my brain. This is the left side of my brain, the back of the brain here. So these are my early visual areas that respond perfectly well to visual stimuli that are not being attended. And then these are the 2 new areas. So this is a real exciting moment as a neuroscientist to be able to find 2 new brain areas. It's a little bit like astronomers finding a new planet or a galaxy. And a lot of people were putting pressure on us about what to name these areas. And some people say, oh, you should name them after your 2 boys, or you should name them after people that were important to you. But and the field is pretty conservative. And so we named them just based on their anatomical location. So they're called IPS1 and IPS2. intraparietal sulcus 1 and 2. This is just where they are in the brain. We're sort of agnostic about exactly what their ultimate function will be, but our working hypothesis, or all the evidence that we have so far, suggests that these have maps of visual field locations, but they're really maps of where the spotlight of attention is located. And so we think that these areas are encoding where attention is directed, and that they send information back down to these early visual areas to highlight those parts of the visual field maps in the early areas that represent the intended location. And that then primes those areas to be able to process stimuli more efficiently. And this is a big part of how attention can enhance perception, is these areas determine the parts of the visual field that are most important, and they tell these relatively dumber early areas which parts of the visual field they should preferentially process. And that's how perception is facilitated by attention. So a lot of work needs to be done to figure out exactly how this takes place. But the fact that there are areas in the brain that have very orderly maps of visual field locations, but only encode attention signals and not sensory signals from the environment, seems to be one important clue in unraveling this. So in studies that we've done since then in my laboratory at Berkeley, we're interested in actually measuring the functional relationships among these different areas. So one real advantage of fMRI is that you can record brain activity from thousands of locations in the brain simultaneously. And so you can say, well, while people are doing a certain attention task, what's the pattern of activity in, say, IPS1, this area that has a map of attention signals, and what's the activity in V1? And is there any interesting relationship between the activity patterns in these areas that changes as a function of attention? So when subjects are attending, is there any change in the coupling between these areas or any change in the temporal differences between these areas? And what we found is that when subjects are sustaining attention at a certain location, the coupling between these areas increases, and that the IPS1 and IPS2 areas are leading the activity in early visual cortex by a few hundred milliseconds. So that also is consistent with the flow of information from these areas down to these areas, that whenever there's a spike of activity in these areas, a third of a second later or so, there's increased activity in these early visual areas. So this is consistent with the idea that these areas are instructing these earlier sensory areas about what parts of the visual field are most relevant at any given time. And so through this kind of analysis, we can develop these circuit diagrams where this is now all the different areas that I've showed you before. And these arrows indicate the flow of information that was obtained from this functional connectivity analysis in our data. And you can see that the pattern overall is that the arrows are emanating from these IPS areas. So these are sources of information flow within this cortical network. And that V1, V2, and V3, these early visual areas, are the recipients of these signals. And so a lot of work remains to be done to really flesh this out and include many other brain areas. But through this kind of method, we can really build a circuit diagram that shows how information moves from one location to another in the brain, and specifically how attention signals filter all the way back down to the earliest parts of visual processing and bias visual perception there. OK, so in the remaining minutes of this talk, I'd like to talk about attention gone wrong. So attention systems in the brain can be injured like any other part of the brain, and there's a very specific and fascinating pattern of symptoms that arises when attention systems are damaged in the brain by head injury or stroke or disease. And one of these is spatial neglect. And so this is characterized by subjects' inability to acknowledge or even perceive objects in the field contralateral to the lesion. So this happens most often with right hemisphere lesions. And so it is an inability to process information from the left visual field. And often, initially, before subjects are fully tested, it appears to be blindness, that people are unaware of stimuli in their field. But this can be dissociated from blindness by a phenomenon called extinction. So if you present these subjects with a very plain visual environment, so a blank computer screen, and you show one stimulus at different locations in the visual field, so you've probably had these kinds of exams in your eye doctor's office, they're able to detect single stimuli anywhere in the visual field. So they're not blind in that sense anywhere. They have perfectly fine ability to detect single stimuli. Where their deficits manifest is when there are multiple visual stimuli in the visual field, which, of course, happens all the time in natural vision. And so if you present two stimuli at the same time, they will successfully perceive the one on the side ipsilateral to the lesion, so on the same side of the lesion. So if they have a right hemisphere lesion, they'll detect paired stimuli just fine on the right visual field, but they'll fail to detect the stimulus in the left visual field, unless it's there in isolation. So it suggests that there's a problem with attention, that they're unable to actually distribute their attention over to the left visual field when there are competing stimuli in the right visual field. And so that's why it's called neglect. Subjects neglect the left side of their visual field. And so these are some kind of anecdotal examples of the kinds of tests that have been done by neuropsychologists of these patients. So for example, if subjects are shown this picture and asked to simply draw it, to copy it on paper, with paper and pencil, they will correctly generate the numbers on the right side of the clock, but fail to generate the ones on the left side of the clock. So there's something about the preferential processing in the intact right visual field that the brain damage has made it impossible to do in the left visual field. Here's another example for a house, successfully replicating the right side of the house, but not the left side of the house. And this is a series of self-portraits that were done by someone who was a very prolific, proficient artist before a stroke that led to neglect. And so initially, you can see there's fairly widespread visual impairments, but extremely severe on the left side of the visual field. And then as he recovered from his lesion, you can see there's these periods where there's always more accurate renditions in the right side of the visual field and less on the left until he achieves a relatively full recovery several months after the brain injury. Here's a way that neuropsychologists can study spatial neglect in a slightly more quantitative way. So you can present people with a paper with all these letters printed on it and ask them to circle every A they see. And they successfully find all the A's on the right side of the paper, but fail to find the ones on the left side of the paper. And again, this is distinct from blindness. So if you were to show them this paper with just this A by itself, for example, they would have no trouble seeing it as an A, correctly discriminating it from other possible letters that it could be, and then circling it. It's only when there's competition from these other locations that you see this asymmetry, this favoring the right visual field over the left.
Is that keeping their eyes on the central line?
It doesn't have to be, actually. So a really important question, actually, one that is being studied by psychologists here in the department in Bangor, is the different coordinate frames, the frames of reference that are used. So for some patients, the deficit is very much tied to the retina. And so if you move your eyes, the deficit kind of moves with the eyes. But in others, it's more invariant. And so regardless of your eye position, you will neglect the left side of objects, for example, and not other. So you could have, for example, two houses, one in the left visual field and one in the right visual field, and you ask subjects to draw them, and they will neglect the left side of both, even though one is to the left of fixation and one is the right of fixation. So there can be these higher order object-centered frames as well, and it depends very much on the details of the injury and exactly what brain areas are affected. Okay, so I'll close with just one utterly fascinating experiment that was conducted by some neuropsychologists in Milan, Italy. So this is an image of Il Duomo, the cathedral in the main square of Milan. And these experimenters had a sample of patients with neglect in the laboratory. These patients had all grown up in Milan their whole lives. They knew the city very well. And particularly, they knew the organization of the piazza, the main square in Milan, very well. And so these numbers here are different shops and buildings around the square. And the experimenters simply asked the subjects seated in the laboratory to imagine themselves entering the square from the south side, and to just report to the experiment whatever they saw in their mind's eyes as they were imagining being in the square. And the patients successfully generated all the buildings on the right side and failed to mention all of the buildings on the left side. So they showed this spatial neglect even though there was no visual stimulus that was being neglected. This was purely visual imagery. They're just in their mind's eye imagining what it would be like to be standing at this location. And then the same patients later on were asked, well, what if you were entering from the north? What would you see then? And sure enough, they've now recalled all the ones that they failed to mention the first time and neglected all the ones that they did mention the first time. And so this kind of neglect, this is further evidence that it's not a perceptual phenomenon, that it's really a higher level attentional phenomenon. Just like we can direct attention to locations in the environment, we can direct attention to visual images in our dreams and in our imagery of environments that we've been in. And so the same systems that are impaired in neglect in terms of having neglect of actual physical objects on the left side of our visual field are also in play for visual imagery as well. So it's this very high-level spatial representation of the environment that seems to be affected. Okay, so in conclusion, I hope I've convinced you that visual perception is not at all like a camera. The eye is very much like a camera, but the rest of the brain processing, which is the majority of our kind of visual experience of the world, of course, we need our eyes for vision, but really why we experience the world the way we do is due to very sophisticated computations in the cerebral cortex. And these computations underlie this active, constructive aspect of vision that's influenced by our spatial attention, our attention to different colors, features, objects, our intentions, what it is that we actually want to extract from the visual environment, and our previous visual experience. For example, assumptions that light comes from above, or that when an object moves behind a tree, it's not actually turning into a different object, it's the same object. All of these things are not given to us by the environment. We have to learn them and then use them to construct our visual perception. I've shown you a little bit about the visual cortical anatomy in the brain. Specifically, the cortex contains many areas, now upwards of 25, 30 areas, each of which has its own map of the visual field. And an important question in our field of research is, why do we need to replicate this map of the visual field over and over again? What are the functional differences in the kinds of computations that are performed within these different areas? And what sort of information do they send to other areas? So even though we have upwards of 30 maps of the visual field, we only have one conscious experience of the visual environment. So somehow, somewhere in the brain, all of the information from all of these different maps and all of these different cortical areas is synthesized into a single unified perception. And we really, at the moment, have no clue about that aspect of visual perception. But we're very interested in studying it. I didn't show you this in detail, but many of these cortical areas have some sort of neural correlate of visual attention. In particular, these two areas, IPS1 and IPS2, in the sort of back upper part of the brain, have maps of spatial attention. So simply by viewing patterns of activity in these areas, you can tell with high fidelity where someone is directing their attention. And where they're directing attention is then being sent back down to the early visual areas where it's influencing visual perception and processing. And finally, I told you about the phenomenon of spatial neglect. This is following brain damage, stroke, or head injury that leads to disorders of attention. And by studying these disorders of attention and how the attention system can go awry, then we get profound insights into the neural circuitry of attention in the healthy, normal human brain. I'd also like to acknowledge my collaborators in the work that I've shown you. So this was the discovery of IPS1 and IPS2 and the analysis of the functional connectivity generating this neural circuit diagram of attention. So my former postdoctoral mentor, David Heeger, who was initially at Stanford University, now at New York University. Thomas Lauritzen was a postdoctoral fellow in my lab at UC Berkeley. He now works at a company called Second Sight, where he's developing prosthetic devices that can be put into the retina of blind people to be able to restore vision. So these devices act like a retina. They absorb light from the environment and transform them into electrical signals and provide some recovery of vision in blind people. And Mark Desposito is my former mentor at UC Berkeley in the psychology and neuroscience departments. And all the research that we do depends very much on the generosity of various funding sources. In this case, the US National Eye Institute, National Institute of Mental Health, and a Swedish foundation called the Lundbeck Foundation. And finally, I'd like to thank all of you for being here on a beautiful summer night and giving me your attention. Thanks.